Hi everyone! In this video, my group will be talking about photochemistry in biochemistry. The topics that we will be covering are EZ isomerization as a tool for biochemical changes in vitro, light and enzyme activity, and photochemical reactions with proteins. To begin, with some background information, photochemical reactions are chemical reactions induced by light exposed to the substance. This light energy is absorbed by the compounds and provides the energy required for the reaction to occur, which is different from other reactions that depends on the temperature. An example of photochemical reaction is photosynthesis. Let's move on to EZ isomerization as a tool for biochemical changes in vitro. We have learned about EZ isomerization in our lectures taught by our lecturers. To recap, the E isomer of a compound is when both its higher priority groups are on opposite sides, while the Z isomer have both the higher priority groups on the same side. EZ isomerization changes the stereochemistry of compounds. Azobenzene undergoes EZ isomerization interchangeably when irradiated by UV light and visible light. In other words, the isomerization of azobenzene between its isomers are not permanent. Photo-induced EZ isomerization of azobenzene is a commonly adapted method of control used in photochromic systems in synthetic molecular devices. This is because azobenzene conjugated molecules are very stable when azobenzene is in its Z isomer form and are not particularly affected by the polarity of the solvent used. Therefore, azobenzene aids in the formation of many photoresponsive biomolecules and phototrigger molecules that control biochemical reactions when highly polar aqueous solvents are used. Let us look at 3 examples to show how azobenzene allows us to monitor the photoregulation of enzymatic activities. We can control and monitor the rate of enzymatic reactions using azobenzene compounds. For instance, azobenzene 4-carboxylic acid, which is covalently bonded to lysine residues of the enzyme papain. By isomerizing the azobenzene compound from an E isomer to a Z isomer by exposing it under UV light, it was found that the rate of enzymatic reaction between papain and its substrate is faster than when azobenzene is in its E isomer form. By switching between the isomers of azobenzene using UV light or visible light, we can observe the photoregulation of the activity of the enzyme. In another enzymatic reaction, isomerization of azobenzene is applied differently. An enzyme, alpha-chymotrypsin, was immobilized in a cross-linked polymer which contained azobenzene molecules. Through irradiation under UV and visible light, the azobenzene molecules in the polymer changed from E to Z isomer and Z to E isomer respectively. When the azo molecules were in the Z isomer form, alpha-chymotrypsin was found to be active, thus allowing enzymatic activity to occur. The activation of the enzyme is due to the change in permeability of the polymer resulting from the isomerization of azobenzene, thus allowing the substrate of the alpha-chymotrypsin to enter the polymer. Lastly, a peptide containing the Z isomer of azobenzene binds tightly to its monoclonal antibody. Upon irradiation with UV light, azobenzene changes to its E isomer, and this caused the peptide to release itself from its antibody. From all 3 examples, we can see that azobenzene can be used differently to control enzymatic reactions. By acting as a coenzyme to speed up the rate of enzyme activity, by changing the permeability of the polymer which can prevent the substrate from accessing the enzyme, and by changing the binding site of the enzyme, making the enzyme lose its complementary binding site with its substrate. What provides azobenzene with this unique function is due to its reversible isomerization, thus allowing scientists to monitor the activity of the enzymes.
Our next subtopic is light and enzyme activity. Before we begin, here's a fun fact on photosynthesis. Did you know that more than half of the world's oxygen is produced by photoplankton photosynthesis in the world's oceans? And 30% of the world's oxygen is produced in our rainforests like the Congo and the Amazon. Light and enzyme activities are best observed in the process of photosynthesis. Light energy is absorbed by chlorophyll molecules and transferred to electrons, which produce ATP and oxygen, which is essential for the survival of animals. However, in some biochemical transformations, the enzyme itself or cofactor is photochemically modified, which may disrupt the final outcome of the enzymatic step. One example concerns photolase, the enzyme involved in the repair of a specific kind of DNA damage. Since photosynthesis is crucial for plants to survive, large portions of the enzymatic reactions are under the influence of light. In the cell, many enzymes that have originated from within the organism can serve as sensitizers, which cause something like an allergic reaction within the cell. For example, hypericin is a well-known sensitizer often found in plants of the genus Hypericum. It has been suggested that hypericin's role is to protect these plants from insect predators. It sensitizes the formation of singlet oxygen, which is in an excited state, and inhibits the enzyme succinosidase. The inhibition of succinosidase hence prevents the formation of fumarate, which will result in a lower rate of photosynthesis and smaller production of oxygen, and eventually plant death.
Next, we'll move on to photochemical reactions with proteins. We'll be exploring these few cases of photochemical reaction in proteins, namely protein DNA and protein RNA cross-linking, absorption of light by specific protein chromophores, and biological photodamage to proteins. To begin with, cross-linking is the process of chemically joining two or more molecules by a covalent bond. Protein DNA and RNA cross-linking can be observed in synthesized reactions with hematoporphyrin derivatives used in photodynamic therapy. In photodynamic therapy, a photosynthesizing agent which localizes the tumor is administered, concentrating the tumor. As the synthesizers are activated by light, a sequence of photochemical and photobiological processes cause irreversible photodamage to tumor tissues. Photodynamic therapy is thus an effective treatment for some cancers. The proteins found in the lens of an eye are susceptible to cross-linking. This results in formation of cataracts. Photosynthesized oxidation of the eye lens proteins may lead to the formation of protein cross-links and high molecular weight aggregates, producing clouding as seen in the photos. Did you know that 78.6% of elderly patients are affected by cataracts in Singapore? The number increases as people get older, so do remember to wear good quality sunglasses with UV protection to slow down the formation of cataracts.
A large variety of photoreactions occurs in proteins when light is absorbed by a specific protein chromophore. A chromophore is defined as an atom or group of atoms responsible for the color of a compound. An example of a chromophore is a tryptophan residue. Did you know the function of a chromophore can be explained with the example of a leaf? When the visible light energy from the sun reaches the leaf, the chromophores in the leaf reflect the wavelengths of the green region. That's why the leaf appears green. Investigation of small dipeptides molecule NH3 and CO2 in vitro showed that there is a loss of NH3 and CO2. Carbon-carbon bond, carbon-nitrogen bond, and peptide bonds are cleaved. Aromatic groups tend to be better chromophores. This can be known by observing the structures of some of the photoproducts. One such example is the tricyclic compound. Dipeptides containing phenylalanine also produces extremely complex product mixtures. The experiment was also repeated with sensitization. Oxidation at nitrogen was an important reaction, and depending on the pH, singlet oxygen was quenched either physically or chemically. The observation of electron transfer into peptide groups support the possible involvement of proteins in long-range electron transfers. The long-range electron transfer has been found in vitro using proteins containing a heme group. To determine if a protein has lost part or all of its activity during exposure to radiation, regardless of the use of sensitizer, tests can be done using an enzymatic assay. An enzymatic assay is a laboratory method to measure enzyme activity. An example of the use of an enzymatic assay is the photosensitization with alpha-tertinyl, which led to the inactivation of acylcholinesterase
and of superoxide dimutase in larvae of the Aedes mosquito. The chemical modifications responsible for such changes in enzymatic activity are not known. Lastly, biological photodamage to proteins can be observed in changes to the skin conditions of those working in outdoor conditions. Photodamage refers to the damage caused by exposure to ultraviolet radiation. The light from the sun, more specifically the ultraviolet radiation, can damage molecules in the skin and DNA. This results in the synthesis of different proteins and enzymes such as prostaglandins and cytokines. The production of these two chemicals lead to the dilation of the cutaneous blood vessels and increase in inflammatory cells, producing the redness, swelling, and pain, or better known as sunburn. This can also result in DNA damage, ultimately leading to the destruction of the skin cell, which is why skin peels after a bad sunburn. Here are some pictures to show all of you what is a sunburn. Do you know that sunscreens employ a mix of organic and inorganic ingredients, such as zinc oxide and titanium oxide, that either reflect, scatter, or absorb the radiation and dissipate it as heat? The ultraviolet radiation absorbed by the skin is thus reduced.
In this video, we have learned the isomerization of azobenzene, a reversible reaction conducted under irradiation. To control enzymatic reaction, azobenzene can act as a coenzyme to speed up the rate of enzyme activity, change the permeability of the polymer, and change the binding site of the enzyme. Moreover, we have learned the importance of light in enzymatic reaction, such as photosynthesis and photosensitization reaction. Last but not least, specific proteins such as chromophores can absorb light and reflect certain colors. Sometimes, absorption of light can result in undesirable protein cross-linkage and photodamage. That comes to the end of our presentation. Thank you for watching.